Well, everyone, we're going to wait just a couple minutes because we got people trickling in. Senna was asking if I would just sing for you all. I will not subject you to that. Although maybe, maybe, I don't know. If it was Cyto, I feel like I might, but it's not happening. And so, plus we'd have to choose a song. What would be the best for Flow? I don't know. Flow goes on, something else. Please. Very nice. Best question we've ever had in a webinar. Full stop. There we go. Now we know. That's it. We got added to the experiment list now. So, all right. I love it. I love it. All right, let's give it about 90 seconds. That doesn't sound like an about, right?
No.
We will wait 90 seconds and then we'll do it. Please no. Charlotte, you can't hear us. I think considering that Safiner just heard my my really bad joke. I think we're good. You're okay. That's okay now. Thank you. Okay, good. She's like, no, please don't sing. That was pretty much what that was. Okay, cool. So actually, as we're waiting, I'll just give you guys a setup for how we like to do this because it makes it a lot more fun. Sen will present. She's going to talk about the NovaFluors and upgrading panels and panel design, and I will take care of Q&A live. I will not sing the answers. I know that's a disappointment as well, unless we get some really good questions. And also I'll take Q&A, and then we'll take some pauses. I'll be managing that, and so we'll just take some time to pause and say, setting, we've got some questions. Let's take a minute and answer those, and then before we move on with the presentation. But it makes it a heck of a lot more fun when we do that. But to intro Sen, before she kicks it off, is Sen earned her BA in chemistry from Williams College and her PhD in medical sciences and immunology and virology from Harvard. She's got over 20 years of experience in flow. She's published over 34 papers and received numerous awards from AAI, President's Rising Stars in Mucosal Immunology, and multiple NIH Fair Awards. More important, at least to me, than all of that is that she's a research application scientist, and she's been the one really pushing the envelope into our 40-color work. And I think even more fun than that is when we sat down for the first time when she was interviewing, I said, this is really going to be like, for a flow cytometrist or immunologist, this is going to be like a kid joining the candy store. And she was like, I don't think so. And so hopefully she can show you what that really means. So with that said, why don't you kick us off? I'm going to stop my video, and then I'll be available for Q&A, and we'll do some moderating.
Yeah, great. I'll go ahead and share the screen. All right. Hopefully you can all see that. All right. So today we're going to really focus on talking about panel design with NovaFluors, so how to really unlock the full potential of your experiments. And the question really starts off with, what if you could design a fluorescent label? What would it look like? You'd want it to be bright and clean, not showing up where you don't want it to be, so silent in the violet and limit cross-excitation. You want it to be consistent and stable. You don't want to use, one set and then find out later, oh, it's a little bit different in terms of its fluorescence because of what antibody it's on. And you'd like to be able to control the brightness. So that if you think about then being able to design that fluorescent label, how does that actually influence panel design? So we're all used to thinking about panel design, but we don't spend a lot of time thinking about the fact that really the labels and dyes are what drive how we set up the panel. And so when you look at things, here we have, spectral overlap. I think Mike has talked about, some of these labels living a much more colorful life, which I really liked as a joke, but also, as a premise, because you can really see, depending on how bright you have to make something, it's going to spill over into a number of channels, whether that's by cross-excitation or just spillover. You often have to look at the antigen density, so, trying to match a dim antigen with a brighter fluor, and then thinking about things, like the staining index and how that's affected by spillover spreading. So really getting that best separation between your positives and negatives. So when we think about dye performance, this is a typical laser setup. These other two lasers and the DUV and IR really aren't used so much, but these 5 lasers are what I'm going to be focused on today, and more specifically looking at some of the blue and yellow-green performance and things that we don't necessarily think about. So this is just showing a typical, what is your primary laser, and you like to think about it being in that channel, but it also, you get some cross excitation. And focusing specifically on Percy P and PE type dyes, the blue and the yellow-green laser often are exciting both of these. And so part of what you struggle with in panel design has to do with the fact that you can't really separate out the cross excitation from these lasers. And so this is just showing an Aurora ribbon plot where you can see that, you've got your primary fluorescence here in the blue channel. But you also have these areas of spread. And so when you're looking at a spectral cytometer, you refer to this as the signature, but really what that is just bad dye performance. You really would prefer to have the dye just in the area that you are looking at and not in these other areas. And you can use these other techniques to get around that, but if you had a dye that just expressed where you want it to, you don't have that same issue. So these are just a few other blue excited dyes. So you can see Percy P has a large amount of fluorescence sort of across multiple lasers. So it's not, although we like to think about it as being, blue excited, you can see that it's yellow, green, and even spilling into the violet and UV. BB515, again, is this, really bright off the blue laser, but there's still some spillover in the UV and violet. And then BB700 has basically cross excitation, plenty of places along the spectrum. So to really address this, we've tried to really look back at how we could design new labels. And so again, focusing on making them bright and clean, consistent and stable, and controlling the brightness. So I'm going to just talk you through a little bit about the platform that we use to do this. And so what you can see here, this is just showing single-stranded DNA, and you have both a donor and acceptor dye, and then this is then assembled into a cruciform structure that we refer to as a phyton. And what this platform does is then allow us to be able to tune both the excitation and emission by positioning and altering the donor and acceptor floors, as well as being able to tune the brightness by building out more of these on each of the arms of the phyton. So this is really the platform that we have, and I'm going to not go into as much data today about building out the brightness or looking at the stability, but we do have other webinars where we're going to focus on that more. I really just want to focus on how you could use this in your panel as you're building forward. And this last is just showing that, the phyton is then bound to the antibody with a single-stranded DNA linker. So what are the labels? So when you look at this, what you can see, what I want to draw your attention to is actually one of the real benefits that I saw right away is it's not confusing how these are labeled. Sometimes I feel like you look up a particular label and you're like, okay, but where is that excited? Where does it emit? Have to do a lot of work to figure that out. So what we've done to try to make that simpler is these are labeled either NovaBlue or NovaYellow. And what that means is that they're excited off either the blue laser, so the 488 laser, or the yellow-green, so 532 or 561 nanometer laser. The second part of the naming tells you where their peak emission is. So in this case, that'd be 530. In this case, it's 570. But what I want to draw your attention to is some of these labels down here, which really are going to make your experiments and the way you use some of these floors stackable. And I'll talk a little bit more about what that means. But just to start out, you see here you have NovaBlue 610 and NovaYellow 610. And for the most part, you can see that their fluorescence in terms of their emission is really similar. But the real difference is what laser are they excited by. So you can see that basically these have a similar type of fluorescence emission, But again, it's where which laser is excited by. And labels like, say, PE are excited by both the blue and the yellow-green laser, so you can't separate out those two things. And a similar kind of thing with the NovaBlue 660 and NovaYellow 660. So in terms of really being able to plan your experiment, one of the things that's most helpful is, again, we have this naming set up so that you know where the peak excitation and emission is. But if you really want to get into the details, there's a great resource known as FPBase, and you can go to that website and look at our labels versus other dyes that are out there, and see both where they're excited and where they emit. So this can help you actually as you're looking ahead at planning experiments. In addition, our labels have been included in the Cytec Full Spectrum Viewer. And so this is what I showed you before in terms of, you can see, have the UV, violet, blue, yellow, green, and red excited. So showing you basically where these are coming off each laser. And one of the things I'm going to draw your attention to is you can see that indeed the blue labels are excited by the blue laser, not the yellow-green, and the yellow labels are excited by the yellow-green laser and not by the blue. Also, you can see that there's very low spillover into the violet. So this makes your designing your experiments a lot easier. So just to get a little more into detail of these particular fluors, what you can see is we have, for instance here, NovaBlue 530, and you can see it's really just, basically emitting at the wavelength that you want. So in this BL1 channel, so this is just showing data from the attune, but it would be a similar kind of spot on your Fortessa or other type of instrument. And the NovaYellow 570, again, very little spillover. And in fact, actually, if you look at all of our labels, you can see that for the most part, there is this excitation just in the primary channel of interest and very little spillover into the other detectors. So looking more closely at how you might look at your panel, this is just, taking sort of a published setup for the attune and a typical one for the Fortessa. And I want to draw your attention to several things here. One, basically the Fortessa often does not have, you have your basically FITSI type dyes and your Percy P type labels in terms of the filter set, and then side scatter comes off of this. But there's no reason you couldn't have an octagon off of this. Part of the reason that the Fortessa has been designed this way is just because of the lack of labels. And since a lot of this you would see from the yellow-green laser, this hasn't been part of that design. But that doesn't mean that that's not something that you could take advantage of as we produce more and more labels. So just to show you here, for instance, PE, you can, again, would see coming off of either the blue laser or the yellow laser. And so, okay, sorry, there we go. Comes off of both the blue and the yellow laser. And you have a similar issue if you look at something like PE dazzle. Again, if you were trying to detect that, you're really not going to be able to duplicate what you're seeing. So you're limited in terms of the experiment that you can do. And then even further, something like PE Sci 5 or Sci 5.5 would also still show up where you have Percy P and Percy P Sci 5.5. So it makes it really difficult to distinguish those. So the thing that we've really tried to do to solve that problem is just showing you on a basic setup. This is where our labels could be substituted, and you can actually get these 6 channels where, in this case, you really could get 2 or 3, really probably 3 realistically in terms of what you're looking at. So you might be able to get a 4th one depending on how bright your other signals are. So, but a lot of what we do in terms of panel design is really, based on this idea of optimizing what can we see versus, when you have this, both cross excitation and this spectral spillover. So this is showing you for instance, the label PE Dazzle. And so that is, it basically has a similar emission to both our NovaBlue 610 and our NovaYellow 610. And this is showing where they're excited by, so their normalized absorbance or excitation. So PE Dazzle, you can see it's excited by the 488 nanometer laser, the 532, 561, which is why you get so much of this spillover both with the blue and the yellow lasers. And again, NovaBlue 610 is excited by specifically the blue laser, but not the yellow-green. And if you look then at the NovaYellow 610, it's really separated from that excitation from the blue laser. So it's really focused on just the yellow laser emission. So this is just a set of single color NovaFluors, and they've been then concatenated into one file, just so you can really see that the compensation is really straightforward, and these are very distinguishable from each other. So just another example of how well that works. So when you're looking ahead at sort of this conventional panel design, you really attempt to even out the positive signals. You want to increase the chance of your separation of the positive negative, but you're always taking into account this cross excitation from the broadly absorbing labels and this spillover. So then the question is, how do we design a panel that minimizes cross excitation and spillover? So I'm going to show you an example. This is actually an experiment I'm getting ready to run this week. And I have put in what, if you tried to use what is out there, this is what you would do. And this is, again, there's no reason that you have to stack all of this into the blue and the yellow lasers. I've got, other things in this panel, but I wanted to kind of show you as a hypothetical. This would be sort of looking at your T cell populations, something with the, you're able to look at CD25 in terms of the Tregs, and then co-stimulatory molecules and PD-1. But of course, you can't really do this. I mean, this is, you first off, PE Dazzle or something similar, you'd be detecting that both off the blue and the yellow detectors. And so you could choose one or the other, but not both. Also, you would have too much spillover likely between Percy P Psi 5.5 and PE Psi 5.5 to really look well at what's going on. So you at most might be able to look at something like this, where you'd have to eliminate two of those. And some of this might be a little messy depending on how broad the spillover is from some of these labels. So if you did the same thing looking at a NovaFluor panel, you can see that basically, because these are either excited off the blue or the yellow laser, now you actually have a minimum of two more, if not more, because some of these, the spillover, it's not just the cross excitation, but also the spillover of the label that affects, how you're able to look at your panel design. So I wanted to just show this as an example. I'm actually in the next week or so, again, I'll be doing this experiment this week or the beginning of next week, and I'll be able to show you the data from that. But this is just an example of, you're looking at your instrument and saying, okay, these are things that I could do. And again, even if you have a 4 Tesla, there's actually no reason going forward that this, you couldn't expand into having, more detectors off the blue laser if you truly separate out the blue and the yellow-green laser. It's just that the instruments have really been built around this idea that, oh, well, we can use, FITC and then PE tandems, and so, we want to limit what we're looking at. So most people can't actually use all the filters that are even on their current conventional instruments. And this is just some previous data also looking at a 6-color TBNK panel versus with our fluors versus a commercially available one. And you can see how much more spread there is into the data, and how you can do this with just the blue and the yellow laser excited fluors. And so the competitor, of course, makes use of both, the red laser as well as the blue / yellow-green laser in terms of being able to separate that out. With photonics, we can just do this with the blue and the yellow lasers. So you can then have other areas where you can continue to expand. And again, as we, generate more labels, there will be continue to be opportunities, but this is just showing you how that would look. And so what you can see, this is just, again, a simple experiment. I'm looking forward to being able to show you the data from that newer experiment, but you can see that our labels actually are very separable. You can see this CD4, CD8 in terms of that separation. Whereas when you look at that with the TB and K panel, you're already dealing with an issue where the compensation, the spillover issue between the cross excitation of lasers is an issue. So you can see that this actually provides a real value in terms of even the panel that you're running now, just being able to expand that. So in addition to looking at conventional cytometers, one of the questions that we started looking at was, okay, well, what does this mean in terms of spectral cytometry when there's all these signatures? Does it matter that we have clean and bright labels, or is that just not particularly important in terms of designing this panel? So what I proposed to do was to look at, Cytec has published a 35 color panel, and they've done, a great job putting that together. And one of those wasn't commercially available. So I suggested that we go ahead and see, okay, what if we take our 6 phyton labeled antibodies and put that in the panel and build out to 40 colors and just see what happens, literally. Does this work, or what are the issues? What do we discover in that process? And this is, really so that we can help, give you that knowledge so that, as you're building out your panels, you can understand sort of how dye performance is really affecting what you can detect biologically. So what you can see here is, this is just showing you the entire panel. And then our NovaFluors are highlighted either in blue or yellow. And so we picked antigens in some cases, such as chemokine receptors, which definitely can be a little harder to separate, as well as some antigen presenting markers and a B cell marker here. So then the question was just what happens with the data? And this is showing you what that spectrum looks like. And you can see it's very crowded. One of the interesting things I like to sort of draw attention to is the fact that even though you've got 34 other colors here, this NovaBlue 610 is occupying a region that really, there aren't a lot of labels that have even really been there. And again, you can see that, you have, this sort of area where you've got a lot of times, say, PE would be excited on from both the blue and the yellow lasers. So in looking at this similarity matrix, you can see that this is looking at the full panel with the NovaFluors and looking at, areas where there may be, issues in the panel. And so the violet dyes are already displaying this sort of bunching in terms of similarity and basically being able to resolve your data. I think there were 10 violet labels in the panel. And so again, adding anything more to the violet area is definitely something that you wouldn't want to do. In addition, you can see here, BB515 and FITC, it's 98% similar in terms of Cytec similarity index. And while you can unmix them, it's very challenging. And the real question is, why would you introduce that into your panel when you could actually have it quite simply easier? So NovaBlue 530 is less similar to each of those than they are to each other. And so we would recommend that you use NovaBlue 530 with either BB515 or FITC alone, because this just, you don't need to increase the complexity of your panel if you're just trying to build out multiple colors. In addition, PE Sci5, basically you can use it in this situation because, again, with the signature, and I'll show you a little more data going forward, that the PE dyes have the ability to be detected off the blue or the yellow-green, and so in a spectral situation, you can switch where they are. But if you're using these on a conventional cytometer, you, again, are just losing out on signal that you can detect. So this is a really interesting plot with the initial 34 color panel. And, as I was looking through the data and unmixing it, one of the things I wanted to verify was, are all of these labels being detected at a similar place to where I'd expect or somewhat different? And if you, basically take this and put it through FlowJo spectral and mixing, it will tell you where it's choosing to look at the primary detector. And so first off, when I saw that the CD45 Percy P, which we predicted to be detected at the B8 location, was being detected off the yellow-green 5, I thought, oh, something's really off. But what's interesting is that when you really look at the spectra, you can see that basically the B8 or YG5 are pretty similar. And so it really is a paradigm shift to think about dyes and labels in this way. We're used to sort of, okay, this is where, this color is expressed, is going to emit, so this is what we're designing for. Well, the truth is, because of all these sort of off-target effects of cross-excitation and spectral spillover, you actually have the ability to detect this other places, but you also need to think about how that's affecting your panel. And it's, it's very important in conventional cytometry, and it still plays a role in what you're able to resolve in spectral cytometry. So then one of the things to really get that information about what's happening here, you can see this is dropping in just one of the NovaFluors at a time and seeing how that affects where the primary detector is. So NovaBlue 530 would be predicted to be detected off the B2 filter. And here it's moved to the B3 because you do have these other two labels that are very specific in terms of the B1 and B2 location. CXCR6 is off the NovaBlue 610, and it really doesn't change anything, nor does the CCR4 off the NovaBlue 660. They are pretty much detected in a similar spot to where you might expect. Where this gets interesting is as you move into these NovaYellow dyes, which are really only detected for the most part off of the yellow laser. And so what that does is sort of push each of the PE or PE tandem counterparts over to detection by a blue laser filter. And when you look at the spectra, again, this makes sense. It's not that there's something different. They actually can be equally detected off each one, which tells us a lot as we're designing conventional panels as well as spectral panels. So you can see here, for instance, the NovaYellow 610 is pushing off PE dazzle to be detected elsewhere. And the NovaYellow 660 is affecting where PE Sci5 is detected. So this is just showing all of that together. But what it really tells you is how, some information about how specific our fluors are in that. In all cases, they're able to sort of push other labels to be detected at places where you might not think of them as being the primary detector. So this is just showing the backbone, a spectral spread matrix. And so what this is telling you as you look at it is where might you have increased spillover, making it harder to detect what you're seeing. So you can see that again, there's some area coming from BB515 and FTSE, some of this violet. So just I wanted to sort of show you that this is part of the original panel. And then the real question is, when we drop our fluors in, how does that affect the spillover, of this baseline panel? And so to do that, again, basically what we're looking at is a delta SSM comparison. So a change in the spectral spread matrix, and looking at just each of these 34 colors plus one to really understand what does that do in terms of the data that you can see. So first off, I've already mentioned that NovaBlue 530 is in a region. So BB515 and FTSE already introduce a large amount of complexity. And this is one place, so I want to draw your attention to the y-axis here. You can see that this is much higher than I'll be showing in any of the others. But really where NovaBlue 530 is still introducing spread is just the BB515 and FTSE. And so again, it matches up what you would predict as long as the label doesn't have, you know, any off-target effects. NovaBlue 610 and 660, you can see, you can drop those in and you have virtually no change in any of the labels with this you know, increase in spectral spread. And further, the NovaYellow 570, 610, and 660, they actually change the spillover. Again, very small amount, because remember the scale is up to 150, not this 2000 that you're seeing on previous. But they introduce a small amount of spread into the NovaYellow 570, being most similar to PE and CF568 to that region, the NovaYellow 610 to PE Dazzle, and the NovaYellow 660 really introduces this change in spillover for PE Sci5. So it's really nice because it's, you know, our fluors are very low impact as you're kind of looking at the panel that you're working with, and you can put them in there and, you know, really go ahead and look at your biology rather than focusing on the dye performance. So what does that mean in terms of your high resolution biology? So as I mentioned, we've already added these 6 colors with marginal impact on the spread, except for the difficult BB515 and FTSE combination. So there's really near zero spread into the other channels. So what does this mean for you? It's easier experimental design, better resolution of biological populations, and easier data analysis. So, you know, all helpful as you're looking ahead at what you might use in your panels. So just to take you through a little bit of the data here, this is a subway plot that Mike put together, and it shows how we did our gating and gives you a way to really look at both the gating of the populations as well as the biology. So he's done this really nice way of visualizing the data. And so what you can see here is you can get a lot of biology out of this. You know, again, there was an artifact here in terms of some of the violet bunching, which isn't surprising, but the rest of the data is really, you know, nice and resolved. And so this is just showing a Tisney plot in terms of perhaps being able to discover new populations, expressing that, and as well as a flowsum plot. So this can really help drive your you know, your panel design and building out to a higher number of fluors, and therefore a higher number of parameters you can detect in your biology. And so we've published this in terms of a white paper that you can download from our website. In addition, this is my favorite place, because when Mike talked to me about, we talked about designing this experiment, you know, I said the thing that most impresses me is being able to look at the data and say, okay, is there some way in which this is, you know, manipulated? You know, what does it really look like? And so that's one of the things we tried to make a commitment to do, was really introduce our data. So you can go to the Phytonics GitHub or Flow repository and download this and look yourself. You don't have to take our word on it. And hopefully that will be a resource that you can use as you're designing your panels going forward. Oops. And so, sorry, my computer's misbehaving. So again, this is just, you know, really the way that, you know, you think about these labels. It really changes how you think about panel design, and that's true whether it's in a conventional experiment, flow cytometry experiment, or a spectral experiment. So as you're building out to more and more, you know, parameters in terms of your biology, you know, we've really shown that this is, you know, these labels are bright and clean, consistent and stable. And again, if you're interested in looking at the brightness, that's something that we can talk about further. So in terms of what we're offering right now, we have a NovaFluor CD4 test kit, and that's available for both either mouse or human. We also have a conjugation kit, and I'll go into a little more detail about how that works. And we are offering custom conjugations for more than 1 milligram of a conjugated antibody. And what you can see here, we are also selling these as individual labels. So if you really look at your panel and you say, you know, it's great for all of that, but I'm really interested in, you know, NovaBlue 610, you can get that. So we can also sell you these as an individual setup. So just to kind of talk a little bit about the conjugation kit. So what it uses is the DNA, you know, from the phyton. So there's an attachment sequence, a unique sequence in between, and then the phyton annealing sequence. So we have this linker that is then attached to the antibody in a simple 3-step process where you basically do 3 pipetting steps and let it incubate overnight and add the quencher the next day. And then basically the second step is a precipitation. So getting rid of any excess single-stranded DNA. And then the last step is similar to, you know, what you do in most PCR reactions. You're basically mixing the antibody and the phyton, heating to 50 degrees, and then slowly cooling that mixture to 4 degrees, and this is ready to use. So it's very straightforward, and you know, you can really get at bigger biology with the antibodies that you're interested in using with this. So hopefully I didn't go, you know, too quickly, but if you have any questions, you can reach out to either Mike or I. And this is also info@phytonics.com is even easier if you just need to remember that. So with that, I can take any questions that you might have.
Great. Thank you, Sid. So while we wait for those questions, I thought I would follow up on the comment about singing and just share with you some greatest hits here. Loving You Is Easy Because You Don't Spread by Minnie Riperton.
Nice.
You Say It Best When You Spill Nothing at All by Alison Krauss. Kiss from a Flows by Seal, another classic song. I Love You Always and Forever, enough said. And also literally my favorite song while we wait for, again, fire your questions away if you'd like to, Ain't No Bandpass Thin Enough.
Nice.
But these are some other hits for you. So even though you didn't sing, hopefully some humor for you. Could we use this, great question, could we use this technology to label MHC class 1 or class 2 tetramer reagents, for instance, instead of conjugating them to antibodies?
Yeah, yeah. So the way the conjugation works is, you know, pretty straightforward. So it's labeling the lysine. So you could label, say, a protein of interest. So I think it makes sense you could label the streptavidin with that and then make the tetramer that you're interested in. So there shouldn't really be any issues with that.
Yeah, it's a great question.
And just so you know, our labels are slightly smaller than PE. And so in terms, you know, that would be the other question you sort of think about is steric hindrance, but PE tetramers and APC tetramers are very popular. So there shouldn't really be any, you know, steric hindrance issues as well.
Yeah, it's a great point too, because, you know, you could do multiple different, right? You could even think around multiplexing in a tube, right? Because you've got the clean fluorescence. So it's an interesting question. Oh, fantastic question. How can we be sure of the standardization of the staining of your dyes, lot-to-lot difference?
Yeah, yes.
So go ahead, Seth. You go for it. All right. So we actually shared the data on this, and so stability fixation, another awesome question. So we actually share our lot-to-lot data, and so I'm going to break your question down in 2 different parts. We have a spec on our lot-to-lot that the actual floors have to be less than 5% on a lot-to-lot basis difference across the entire spectrum. right? And so we actually take that data and actually, so we put it in our white papers, Seth's probably pulling it up now in a PowerPoint. And we also test that in flow cytometry, both in our lab and in an external lab. So the CVs on that are 4% or under on a lot-to-lot basis. It's the first part of the consistency piece of this. And here's that data. And so if you look at the top plot, you're actually getting less than 3% off one of the NovaYellow labels. And then on the NovaBlue here, you have one label that is still under 5%. All the other ones are literally bang on across the entire spectrum. And because we're reliant, if you think about it, we're relying upon oligo manufacturing to do this. So it's incredibly consistent what we're able to do. The second point to make there is that when we make a label that has, for instance, fluorophores and component dyes only off 2 of the 4 arms of this phyton structure, 100% of what is in that label is just that, right? And so that's another part. The other part around the standardization that I think is equally as important is every new batch of label that we get and that we create, we QC on PBMCs for every single lot. So not only are we saying we want to maintain the manufacturing consistency, but we also put that on antibodies and make sure that that is in fact staining on cells and the CVs are low, right? So really I would say it's a great question because one of our goals is to try and make this lot-to-lot variance really a thing of the past. So hopefully I answered your question and hopefully a couple different levels of that question as well, because it's a really good one. What about the stability to fixation? Yeah, so could you full screen that, Seth, and then maybe you can take that one.
Yeah. So this is just showing you, we like to refer to this as the absent-minded graduate student experiment, and I've certainly worked with people where their intentions were good, but something gets left out on a bench. And so this is just showing you, we are showing for up to 2 weeks, but we've actually left this out up to 3 weeks, and there's no change in the fluorescence there. In addition, when you look at fixation, so this is, stained in this case with a NovaYellow 610 labeled with CD3. And even if you wait 14 days afterwards with fixation, there's really no difference that you can see between day 0 and day 14. So yeah, so our labels really function, although we make use of fret, they really aren't, similar to a tandem in that, it is controlled because of, this sort of solubility cage with the phyton.
Great. Next question. More questions coming. Fantastic. Keep them coming.
Yeah.
What is the efficiency of the conjugation?
So our partner has said that they are over 85% in terms of what they have published. So.
Yeah, and we've seen it be 60 to 80% in our hands on the final step there.
Yeah.
So how about the stability at different staining temperatures? For example, chemokine receptors sometimes need to be stained at 37 degrees.
Yeah. So our labels are incredibly stable, so there really shouldn't be any issues with that. I mean, again, even when we're setting up the initial annealing between the antibody and otherwise, that's done at, at 50 degrees and then cooling to 4 degrees. So we literally do that experiment every day. there's no issues there.
Maybe I can take the next one because
Sure.
It's how to make the subway tree. It's very visual, any special software. Very timely question because I was actually just writing up a Medium article on how to do this. So the software I use, because I couldn't find a good software tool for it, is I actually use PowerPoint. So we freely share that PowerPoint template as well with folks. I may or may not be twisting arms to try and get this into a software package. But I'm also writing up something on like what makes a subway plot work, for self-phenotyping. So hopefully put that up today too on LinkedIn. But yeah, happy to send that your way. Another question here. Are your dyes resistant to fixed perm solutions? Are they stable?
Yeah. So we have tested both formalin and paraformaldehyde, and this data that I'm showing you right here is actually 14 days with, I think this is 1% formalin in this case, but we also have data from 2% paraformaldehyde. So yes.
Yep. And actually, I'll just add one more more part to that, which is that, you know, we think that the structure, the cartoon of this phyton looks like a flat structure, but it's actually curved. And so we think that that adds a lot of stability. I mean, we've seen that it adds a lot of stability to the platform, and really provides it the solubility cage, essentially, because it's binding water to the DNA itself, right? Which is why we see some of these really interesting results with stability. Next question, and we have data on this as well. Can you guys produce fluorochromes that are specific for cytometers, such as the Symphony A5?
Yes, yes. We are continuing to build out the spectrum, so I would say stay tuned. There's one start of that. So in June, I think we already have it on our webinar list. So keep tuned for any of the new fluors. But yes, you can absolutely use this on a Symphony, and we actually have testing data. I'm going to see if it's on here, but literally
it is in there.
Yeah, there
it is.
Yeah. So this is just showing all the comparisons that we ran in terms of the Symphony. So, basically not just, the direct comparisons, but other labels, just so that we would have that data. And so this is showing you the NovaBlue 610, which again, as I pointed out to you, it's almost like weirdly alone in this spectrum of, what else is actually out there. So the Brilliant Blue 630, you can see is a bit brighter than the NovaBlue 610, but it introduces a ton of spillover. So this is showing you that right here in blue is our NovaBlue 610. And then we also have the comparison for the NovaYellow 610 with a number of comparator fluors. So again, sort of the CF594PE, Alexa fluor 594, and ECD. And again, our label really introduces very little spillover and is quite bright. And so this is just showing the separation index. So the separation index is a way of sort of thinking about, how much are you separating the positive and the negative, and sort of quantifying that, because if you have a lot of background, then it doesn't matter that you have something that's bright. So you can see that these two labels are similar in terms of their separation index to APC. And Steve Perfetto at the NIH, who ran these particular experiments on the Symphony, actually found that for the NovaYellow 610, it contributed less spread than any of the other comparators measured. And so
yeah, and there were two follow-on questions that I'm going to try and put them together, because it really is honing in on, essentially the differences, or essentially the replacement, if you will, of NovaBlue 660 and its comparison to this Brilliant Blue 630, mm-hmm. right? And so I think if we were to walk back through this and say, it is less bright, but what's the cost of doing business of Brilliant Blue? And the answer is, as I said and showed you, a lot of spill, a whole lot of fluorescence in the violet, right? And so that's, I think, hopefully combining those questions correctly. Could both of those, I think there's another interesting question for you, Seddon, which is, could you, for instance, use Brilliant Blue 630 and then NovaBlue 660 together if you were running this on a spectral cytometer?
Yeah, so you can run a lot of things on the spectral cytometer in terms of, because again, the signature means that you can detect, some of this other places. And since we already know that the Brilliant Blue 630 has a lot of spread, that can be taken advantage of. On a conventional cytometer, of course you have to worry, because you have to detect it, at the place you're detecting it.
Yeah. Any idea about compatibility with oligonucleotide conjugated antibodies? So I'll for example, if you stain your cells with AbSeq or TotalSeq antibodies, but also need to stain with fluorochrome conjugated antibodies to sort before sequencing. I'm sort of like begging to answer this question, if that's all right.
Please, yes. I was like, I feel like you want to.
Man, I'm dying, because because one, it's a good question, but two, it's also because the answer is absolutely. And so the conjugation kit relies upon a different oligo-oligo interaction. Well, the stainings that you're seeing, for what it's worth, on a lot of the flow that we have run is us using a poly T tail onto the phyton, and then using the reagents that you're mentioning, right? And so, you can actually add these. So what you can do is actually add in the poly-T labels right into your antibody tube, right? And then you can go and stain your cells with those totals. So you can run them on flow, you can sort them, and then you can sequence them. So there you go.
Yeah.
Cool. Another one. Is it okay to conjugate a phosphoprotein to a NovaFluor? Might be an antibody, I think it might be the antibody to phosphoflow. I'm not sure if that's the question, to make sure. But that one's for you.
Yeah. Yeah. Well, first off, I mean, based on anything we've seen, you know, this is really just a standard conjugation in terms of protein chemistry. So targeting the lysine. So whether that's on an antibody or, you know, another protein, that shouldn't be an issue. We just haven't actively tested the phosphoflow fixation. We're actually looking to do that soon. When I give these webinars, or Mike gives a webinar, I actually have a running list of the experiments that I'd like to run. So this is definitely high on the list of things I'm excited to go into the lab and test out. So.
Cool. We'll hang out for another couple minutes here, but I'm glad you finished early, so we had time to cover all the questions. These are great. Here we go. Do you have a catalog for cellular markers, for instance, CD33, 11B, TREM2, conjugated with your fluorochromes, or do they have to be customized or custom ordered?
Right. So right now, we're just selling the conjugation kit. But again, I would state, keep your eyes, you know, peeled. We are always looking to build out what we can do.
Yeah, I'll add one part down to that too, which is that we've had a lot of interest in custom conjugations that are a meg and up. And so if that's something that's of interest to you and to support that, you know, happy to discuss that and discuss what that means.
And just to follow up to that in terms of the conjugation kit, you know, you were asking about, this combines the question on the Symphony and otherwise, but we have a customer that was asking about, you know, how could he upgrade his panel? And I spent some time looking at that, suggested some places where there was actually room in his panel to do that. And then even, you know, suggested that he send me, you know, any antibodies he was looking at. So I'm happy to do that if that's something that you're looking at, to say, yes, that would be, you know, a good clone to use, or maybe you want something a little more concentrated. So mostly what we try to do is have it be higher than a mg per mil in terms of the antibody that you're using, and not to have other proteins, because guess what? Then you're labeling just the other proteins and not your antibody. So those are the main things.